I actually got into this sort of special thing. I should be very quiet today. So it is all Om and Swastik and everything. आपको दिखाई दे रहा है या नहीं?
Yes, we can see all the Om,
Yeah.
sir. I hope they work.
I was debating because I have another T-shirt which is very nice. Its printing is ki persona non grata.
This is better then.
Yeah.
It's a very nice one, actually.
It's
Yeah,
9.
this
Can we start?
Yeah, we start straight away.
Okay.
Okay, Archana ma'am,
Yes.
be on standby. We'll be going live on the count of 5. Okay, so all the members in the meeting to be on mute. Thank you. We are going live. 5, 4, 3, 2, 1. We are live. Go.
Good evening, everyone. Welcome once again to the Wednesday Cafe from iTech. It is a pleasure to be here and bring you the best of faculties from all over the world and our own country. We are doing a series on pre-diabetes, and we are looking at the science, the philosophy, the evolution, the pathogenesis, and the various drugs that work at that level. Today, we have amazing speakers, and the first of whom is Dr. Chitrajan Yagnik. So I will be introducing him and then welcoming him for his talk. I know that everyone knows him. He does not need a formal introduction, but still, the kind of a research person we have amongst us is something we must know. He is the professor and director of Diabetes Unit, KEM Hospital and Research Center, Pune. He is attached to IISER in Pune and a visiting professor in molecular medicine, Exeter, UK, and also a visiting professor in Danish Diabetes Academy. His research interests are of high susceptibility of diabetes in Indians, intrauterine programming of diabetes, and vitamin B12 and 1C metabolism. So these are some things, the research is something that we learn from Sir. We are always inspired. He always scolds us and guides us to go further when it comes to research. With innumerable awards to his name, from way back of 2009, the Helmut Mernet Award from the International Diabetes Federation, to the recent 2019 March of Times Agnes Award. So his publications are 293, first author in 78, and these are like something which is overawing, and that is why we want him here, and he is going to take us through the evolution of pre-diabetes. Over to you, sir, Dr. Yagnik, sir. Sir, you are on mute, sir.
Right. Thank you, Archana. Thank you, IDEC. Delighted to join you. And I remember the discussion we had at the last meeting with Professor Ralph DiFronzo, Anup, and Ramu. And I think I was really waiting to show some interesting results of our research, which would put some new thoughts on how the pre-diabetes evolves. Also, the point I raised that what the speakers in the last meeting, including Dr. Ralph DiFronzo, was saying was actually pre-diabetes contaminated by obesity. And therefore, there was a lot of concentration on obesity-related phenotypes and biomarkers. And I think after spending 30 years and setting up a birth cohort in the village, I think I'm going to show you what might be pure pre-diabetes without contamination or much contamination from obesity adiposity. So let's get into it. And I suppose you are able to see my slides. Yeah. So I called it Journey to Pre-Diabetes because there is pre-diabetes, there is pre-pre-diabetes, and I'm going to tell you there is pre-pre-pre-pre-diabetes. And these are the learnings from the Pune cohorts. I come from the KEM hospital. Very proud to say this is the only diabetes unit in the world where mother and baby is the logo, long before even I met David Parker. The new logo we have put for our diabetic patients is the modern Ganesh, who instead of traveling now on the back of the mouse, which he used to do when we were children, now he clicks the mice of the computer and actually moves very fast. And that's the rapid transition we have. A combination of these factors has caused an explosive epidemic of diabetes and other non-communicable diseases in India. You're always supposed to declare your conflicts. I did my confessions 5 years ago in European Journal of Clinical Nutrition by saying that I am not any of these things, either an epidemiologist, nutritionist, pediatrician, obstetrician, or body compositionist. But I like to investigate these. And today I am going to put up a new confession that these are the things I am not, but I am a neurotic, compulsive, and antisocial person. But in scientific and statistical terms, basically I'm normal within the 95th or whatever the confidence intervals you want to have. So natural history of diabetes, as we know, is that it starts with, like, say, a normal population where we don't know whether, basically they are normal glucose tolerant. And then finally, we are interested in diabetes. And when I was a student, there was normal glucose tolerance and diabetes, but sometime in 80s, the WHO committee introduced the concept of pre-diabetes, which generated a lot of interest, lot of controversy. We are not going to go into that. But the diabetes prevention trials then decided that conversion from pre-diabetes to diabetes was an important outcome, which they could use in the prevention trials of diabetes. Now, as you can understand, life is a continuous thing. So pre-diabetes is only early diabetes. And the cut points may be debated. But we are not going to again discuss that. We are going to use either ADA or IDF WHO cut points. What I've shown is that there are arrows even before NGT. And that's what actually we'll discuss later on. And of course, there are a lot of diabetes complications and outcomes after diabetes. So the diabetes prevention trials, and I've put 3 of them: the diabetes prevention program in the US, the Finnish, and the Indian. They treated hundreds of thousands of people who were diagnosed with pre-diabetes by glucose tolerance testing. And they were also obese in the American and the Finnish trial. In the Indian trial, they were not obese by the BMI criteria, but they had a higher BMI than the non-diabetic or non-pre-diabetic people. And as we know, as we have described from my department, at this BMI, we have much more body fat than the European people of similar BMI. So I have always said that we call this primary prevention. And what are we really doing is we are selecting middle and older age people with obesity and glucose intolerance. So all these 3 are end stage factors. And then we are saying that we are going to prevent diabetes, primary prevention. The advice which is usually given is eat less and walk more, or eat anti-diabetic drugs. Again, that shows that we believe that their glucose intolerance can be treated with these tablets. So I have objected to this being called primary prevention. It's a treatment of the condition. All these ideas, when I was learning as a young diabetologist, were actually put to a toss when David Barker came to see me in February 1991. And he showed me these results, which are yet to be published in BMJ. In a couple of months, they were published then. And this was very surprising because I was trained as a diabetologist and was weaned on the idea that large babies born to diabetic mothers are at higher risk of diabetes. What David Parker showed in Hertfordshire cohort was quite the opposite. He showed that lower birth weight increased risk of diabetes and metabolic syndrome. And actually, there was no low birth weight. It was always a lower the birth weight, higher the risk. So it's a monotonic increase. It's continuous. And again, he was very careful to say this, that this is a continuous phenomenon in the population. I can also tell you that I suggested to him in the first meeting the nomenclature thrifty phenotype to him. I suggested that you call your hypothesis thrifty phenotype, which he did. He first called it fetal origins of adult disease thrifty phenotype, and which he published in a couple of years, where he said type 2 diabetes is the outcome of the fetus and early infant having to be nutritionally thrifty, suggesting that that fetus was undernourished in pregnancy, as opposed to then prevalent idea that overnutrition of the fetus produce or increase the risk of diabetes in later life. So that time there was quite a bit of discussion and everyone said this must be genetic. The genes which produce low birth weight must also be associated with either insulin secretion, resistance, etc. And therefore, it was actually a common antecedent. This, of course, could be true. I'll tell you how it might be true and how it might not be true. But yes, that was a possibility. But very soon, actually, David Parker suggested that this was epigenetic rather than genetic. And this was a concept which was developed by a British embryologist, Conrad Waddington, in late 1940s. And I read this as a BSc student in 1970. And what he said was that once a fertilized ovum is there, it has all the genetic factors which it has, which it passes on to every cell after division. But from that single cell, we have a whole lot of other cells, organs, systems, and the individual which develops. And therefore, he said that from this genome to the phenome, which is the newborn infant, there is something called epigenome, something over and above the genetic structure of that cell, which converts it into different phenotypes. And he said environment drives this. And that time, of course, they didn't know how or when. They had extreme animal experiments to prove that. But over years now, of course, the concept has widened quite a bit. And then David Barker's theory got converted into DOHA, the developmental origins of health and disease. And the basic idea is that maternal influence on fetal growth, epigenetic influence, reflects in its growth, development, and differentiation, which can be measured at birth as certain phenotypes. Low birth weight was a very common sort of phenotype based on birth weight, but now that has been refined to so many things. And I added the body composition to it. So in fact, baby was actually part of this. And there are so many other biomarkers. And then subsequently, postnatal growth also comes into it. That's again epigenetically modified. And then we actually can measure risk factors, morbidity, and finally mortality. So it's a very continuous process starting from conception or before right up to the end stage. And the central concept is programming. And programming means actually affection of the blueprint of the development of that individual. And this is now known to be reflected in what we call multi-omics molecular structures, cellular, tissues, organs, systems, and organism in various ways. And it is by effect of various, like say, environmental factors, adverse factors, which restrict the plasticity of the developing organism. These are very standard concepts in embryology, but what we are basically saying is that the full genetic potential of that development is actually curtailed if there is an adverse environment. Then came the concept of first 1,000 days to include the pregnancy, lactation. And we know that these stimuli which actually do this can be nutrition, maternal metabolism, stress, pollutants, etc. And a very important thing to know about development is that development promotes growth of brain. It protects the growth of brain at the cost of other organs. So if there is an adversity, there is a brain preservation. So that as if brain is saved to maintain the identity of that species. And this is done by adjusting the circulation so that the pre-ductal circulation in the fetus is promoted while the post-ductal circulation suffers. And in the post-ductal circulation, you have heart and its circulation, you have lungs, liver, muscle, kidney, pancreas, and the lower extremities. And as you can well imagine, today's non-communicable diseases are all referring mainly to these organs, and therefore an adversity in utero challenges the development of these organs, which in turn predisposes them to problem. That's programming and with brain preservation. Remember short legs because I'm going to tell you something interesting about it. So finally, I realized that life can only be understood backwards. This is the Danish philosopher who taught this to us. I became quite involved in this when I became visiting professor to Denmark. And that's a fantastic thing to understand. So based on that thing, we decided to set up a prospective birth cohort in Pune. Dr. David Barker and Caroline Fall were my colleagues in this. And in 6 villages in 1993, we screened the whole population and enrolled 2,675 women who were married but not pregnant. We followed them regularly, measured the anthropometry, hemoglobin, and nutrition. 814 of them became pregnant. They were intensively studied 2 to 3 times in pregnancy. Babies delivered, they were measured in detail at birth. Then they were measured every 6 months for 18 years and then every year. And every 6 years, we also do a very comprehensive measurement in the baby and the parents related to their size, body composition, nutrition, cardiometabolic and neurocognitive factors, etc., etc. The list is increasing. And 24-hour follow-up had to be stopped in March 2020 due to COVID, but we have started from this 1st April. And we set up a biobank from the parents and the children serial biobank, which is envy of so many others. Out came, like say, the first major finding from this, where we described the thin fat Indian baby, where I compared the measurements of Indian baby with an English baby and showed that though Indian baby was 800 grams smaller, it had more fat. It had higher leptin and lower adiponectin concentrations in the cord blood. And that was quite an interesting and stunning finding, that what looked like a thin crony baby had actually deposited more fat than a healthy looking English baby. And the cord blood actually showed the biomarkers of that. Everyone again told us this is genetic. It took me almost 25 years to prove what it might be, and recent publication in journal Diabetes from the GIFS consortium, which was led by us, we showed that for same gene risk score for birth weight, which was developed in Europeans, the Indian babies are much smaller. So for each gene risk score, the blue babies here are the European babies and these others are all South Asian babies. The Pune maternal nutrition study forms the largest part of the South Asian babies. So despite having similar genetic risk score, Indian babies are smaller. Answer is that it's driven by environment. What happened subsequently? I mean, there's a big story in between, but I'm going to speak on pre-diabetes at 18 years. So at 18 years, 29% of these offspring had pre-diabetes by the ADA criteria. In boys, IFG was more common. In girls, they were similar, IFG and IGT. Two girls also had diabetes, and at the same time, they were nutritionally also deficient. So B12 deficiency was present in 50 to 60%. Pre-diabetes was more common in males, 37%, as opposed to females, 20%. So you can roughly say 30%, 40%, and 20%. Twice as common in boys. And if you look at their average BMI, the hyperglycemic, that's pre-diabetic, had an average BMI of 18.9, NGT had 18.5, no difference. And that's why I call this pure pre-diabetes, as against what you will hear from everyone else, where obesity is the major driver factor. And I think that actually is a confounding and a contaminating factor in thinking about pre-diabetes. And actually, 1/3 of pre-diabetic boys and 2/3 of pre-diabetic girls were actually underweight, BMI below 18.5. And only 8% were overweight. Even measuring body fat by DEXA, only boys had marginally higher body fat percent. Girls had similar body fat percent compared to the NGT girls. And then because we had this information going back 30 years in the parents, in their own measurements, and mothers from even before pregnancy, we were able to find out what are the determinants of pre-diabetes at 18 years. There were some related to undernutrition and some related to overnutrition. This is very interesting. This is the double burden of malnutrition, which is driving pre-diabetes in this population. The undernutrition measures where they were short at birth. And that's why I wanted you to remember about the small legs in the challenged fetus. They had small head circumference. That means whatever challenge was there had actually also reduced brain growth to an extent. And the mothers were non-overweight. The overnutrition type of factors which are associated, that maternal pregnancy glucose was a determinant. And remember that in that population, out of 700 women, only 3 had marginal IGT. Otherwise, they were all normal by the current criteria. But higher glucose within normal was a predictor of higher glucose in the child. Paternal glucose intolerance was a predictor for daughters. So there was actually a separation of parental influence on the offspring. Mother had influence on both boys and girls, father had influence on daughter. So there's a combination of parent of origin effect and sex specificity. And males had higher adiposity, as I showed you in the previous slide. So this was quite a revealing finding. It's published in Diabetes Care 6 months ago. So then I had developed this concept about 15 years ago of dual teratogenesis, drawing from Norbert Frankel's idea of fuel-mediated teratogenesis in a diabetic pregnancy, and the undernutrition coming from David Barker. But Andrew Hattersley also showed that genes which influence insulin secretion can also affect the birth weight. So the current story is that lower insulin concentrations in the baby, probably because of poor pancreatic development, either due to nutritional insults and in very rare cases because of genetic defects, leads to higher risk of adult diabetes. And this is the story of the low birth weight, while this is the story of the large birth weight. And in a country like India, there is a combination of the two, especially when we move from rural to urban, and that's why I called it dual teratogenesis. We had studied incident hyperglycemia, that's pre-diabetes and diabetes in the mothers 6 years after the delivery of their child. And what did we find? Lower leg length in the mother was a predictor of pre-diabetes and diabetes 6 years after delivery, higher fat mass, higher total leukocyte count, and weight gain from pre-pregnancy to the follow-up period. So again, it is a combination of early life deprivation and subsequent overnutrition and some inflammation. And on this background, I have to tell you that in last 150 years between 1830 to 1980, when Europeans gained 15 centimeters in height, Indians shown here in the yellow line gained 0 centimeters. So we have had a height deprivation going back 1.5 century up to 1980, after which things have improved. So diabetes is a combination of secretion problem and resistance. And with the evolutionary biologist Jonathan Wales, we wrote a paper in 2016 about capacity and load model. So we said in Europeans, the capacity due to lean mass, higher lean mass and lower fat, as opposed to Indians where there is a lower lean mass and higher fat mass, there is an imbalance between lean structures and fat. And one of the lean structures is of course pancreas and the islets, because it's the derivative of the lean mass. And if there is low capacity and subsequently high load, either because of adiposity or nutritional, then that can lead to problems. Another thing I had done 30 years ago was to measure fetal organ development in utero, and dramatic results were seen for liver growth. Children whose liver grew poorly, that's the lowest tertile of liver growth in utero, but who had a larger liver size at 18 years measured by ultrasound, they had the highest prevalence of prediabetes. And you will see prevalence of prediabetes was highest if you had poor liver growth in utero. This is the first demonstration anywhere in the world of intrauterine organ development defect as predictor of a metabolic abnormality later on, and also supports what we had shown before, that if you're born small, that means if you have small organs, but later on grow big, then there is trouble. And in boys, there was lower insulin sensitivity, but HOMA beta was similar to those who were NGT. In girls, HOMA S and HOMA beta both were similar. And this is a problem with this model, that you have to difficulty interpreting this sometimes. So you have to interpret HOMA beta against the prevailing HOMA sensitivity, and that's what we call disposition index. And you will see here disposition index was lower in pre-diabetic boys and girls in both, and that problem was bigger than the sensitivity or the HOMA beta issue. So lower insulin secretion is more prominent than insensitivity, especially when we combine the two like Bergman did in this disposition index. And then we trace the life course evolution of glucose and insulin in these. So red lines are those who became pre-diabetic at 18 years. If you look at their glucose levels, they were higher at 12 years and they were higher at 6 years. So those who were pre-diabetic, diagnosed pre-diabetic at 18, had higher glucose right from childhood. Insulin concentrations were no different, but only at 18 years. HOMA S actually came down with increasing age. HOMA beta tried to increase to compensate that. But when you put the two together in the equation, we found that the disposition index of those who had pre-diabetes was lower not only at 18 years, but also at 12 years and also at 6 years, compared to those who were normal glucose tolerant at 18. Again, first time demonstration of going back into childhood in a serial measurement in a cohort with more than 90% follow-up rate. And then we found actually this analysis to see what is the predictive value of fasting glucose at 6 years of age and 12 years of age for pre-diabetes at 18 years of age. And higher fasting glucose, that's in the highest quintile, predict or increase the risk 3 times for having pre-diabetes at 18 years, and having higher glucose, that's in the highest quintile at 12 years, increase the risk 6 times. In other words, there is a strong prediction of 18-year pre-diabetes by fasting glucose at 6 years and 12 years. And this is a very important finding. This could be put into public health practice. Then, of course, the girls in the trial, girls in the Pune maternal nutrition study have grown and they married and became pregnant. So we have now data on 200 pregnancies in the F1 generation and on the F2 generation. So we did a glucose tolerance test in pregnancy in these girls, and you want to see what was their tracking. So you can see here that fasting glucose of girls who had highest fasting glucose in pregnancy was, here at 28 weeks gestation, was already higher at 18 weeks, 18 years, 12 years, and 6 years. So like what I showed you for 18-year pre-diabetes, for let's say pre-diabetes in pregnancy, they had higher glucose tracking back into their childhood. And similarly, their disposition index was lower at 18 years, 12 years, and 6 years, while their HOMA S and HOMA beta were no different in childhood. In other words, what we found for prediabetes at 18 years is very true for gestational glycemia. And this tells you that actually gestational glycemia is not really gestational. You have heard me many times that gestational diabetes is not gestational. And quite provocatively, we don't know what is diabetes in pregnancy. This was said very nicely by John Jarrett in 1993, who said GDM is a case of glucose intolerance, who is also pregnant. And that's what, for the first time in the world, we have this serial data. So pre-diabetes in pregnancy, what we call gestational diabetes, track back to 6 years of age. In other words, that girl's ova from the childhood are exposed to abnormal milieu, and periconceptional programming is going to be affected much before you diagnose GDM. And this is very similar to 18 years. So measurements of fasting glucose increase the risk of gestational hypoglycemia many times. Are there any epigenetic markers we can diagnose? We are working on DNA methylation. We are working on metabolome and proteome. But this is the first paper we published last month in Journal of DOHAD. It's about miRNAs. Circulating miRNAs. miRNAs are one of the epigenetic mechanisms. And what we showed was in the girls, because we didn't have money, we just chose 10 girls who were pre-diabetic and 10 who were normal. But even that gave us a fantastic information that the miRNA differential expression in the pre-diabetic girls at 18 years actually tracked back to 6 years of age. So miRNAs in the circulating blood were different even at 6 years of age. And we are now expanding this measurement to the full cohort. So we will have data on many hundred. We will see how it holds, what additional markers we find. And there are some interesting markers which actually told us that TGF-beta pathways might be affected by these miRNA disturbances. That's a developmental pathway. Another story is in 2017, Indian government published what is called CNNS, Comprehensive National Nutrition Survey, which based on our prodding, actually collected fasting blood from children between 5 to 9 years as well as 10 to 19 years. And this map of India shows you in increasing pink colors the prevalence of pre-diabetes in these young children and adolescents in different states. And dark red, you will see, is more than 21%, even in children from 5 to 9 years. So this vindicates what we have found, and this actually raises an alarm. And I think this is something which we need to take into account in our public health policy. We are also very active in GDM. That's another topic, but just to show you that follow-up of 200 children born in diabetic pregnancies shows that at an average of 15 years, 5% of them were diabetic, 37% were pre-diabetic, as opposed to children born in non-diabetic pregnancies, 20%. And even those below 10 years where we did not do glucose tolerance, their glucoses random of fasting were higher than those children born in non-diabetic pregnancies. So again, this is a sort of fetal overnutrition story. So now I can tell you that actually there are very early stages, and therefore diabetes prevention trials somewhere here, I think, is actually after the horse has bolted. And I contributed to a Lancet sort of consultation where we called it begins before the beginning. And this is the primordial prevention, which usually starts intergenerationally in the mother, but there is now increasing data from epigenetic contribution from the fathers, especially Elizabeth Patty from Jocelyn. And therefore, we have to really think differently. And I have shown undernutrition of the fetus as a major driver of this in our cohort and obviously across India in many states. And in 1988, when Wellcome Trust gave me the first grant, they agreed to do a meeting on MRDM, which is malnutrition-related diabetes. I was supposed to be the first speaker because my grant had driven it, and I showed this slide. Englishmen like to say, rain, rain, go away, little Johnny wants to play. In India, आपण म्हणतो ये रे पावसा, तुला देतो पैसा. Or you go to Kalidas and he will talk of Ashadasya Prathama Divse. So extremely different concept coming from opposite side about monsoon here and again for diabetes. And Dr. Tripathi had really written this seminal paper in journal Diabetes in 1965, putting hundreds of patients in Orissa as malnutrition related diabetes. And 6 months ago, we published the subtypes of type 2 diabetes in type 2 diabetes diagnosed before 14 years in my department and in other places. I'll show you in collaboration with LEF group and Rashmi Prasad. Rashmi is in Pune today, actually. So this was the group. And what we found was in Sweden, 67% of type 2 diabetes diagnosed before 45 years age were MOD type, which is moderately obese diabetes. While more than 50% of people in our cohort were SIDD, that is severely insulin deficient. Then I got 2 more cohorts as a validation from Ahmedabad, Banshi Sabu, and from Dibrugarh, Sanjeev Kokoti and Anupam Gupta and Anupam Datta. And as you can see, Ahmedabad and Pune not very different. But in Assam, amazingly, 67% of young diabetic patients were severely insulin deficient. Again, suggesting that strong environmental adversities, probably acting early in life, actually drive insulin deficient type 2 diabetes. This is a cartoon I drew many years ago, just to say that what we study as end stage actually starts much, much earlier through genetics and epigenetics in utero, then early phenotypes, late phenotypes, etc., etc. And if you study here, then the horse has already bolted. So we can never understand the pathophysiology by end stage because there's a lot of reverse causality also. And the epigenetics is evolving throughout the life cycle. Very interesting. So in summary, I'm going to say, I've shown you story of what I call pure pre-diabetes, uncontaminated by obesity. And that has a different connotation. And that is what must have happened even in the Western populations a few hundred years ago. Diabetes is an intergenerational life course evolution, so DOHAD. A primordial epigenetic modifiable component is there, and that's very important because we could do something about it. Metabolic abnormalities are detectable early in childhood, and again, we can do something there. There is a progressive accumulation of risk, and public health policy should concentrate for prevention of diabetes on fetal, childhood, and pubertal growth. There should be a childhood screening to implement various strategies. And adolescent health is very important because it's intimately linked to reproduction and effect on the next generation. And I have also shown you by subgroups that treatment strategies in young T2D patients in India should take into account the severely insulin deficient. Mohan published a paper after we published, it's called Inspire. So I'm sure he was inspired by what we had written. And he showed genetically and phenotypically, again, same thing, that in young diabetics, insulin secretion was a major defect than insulin resistance. This is a film I always like to advertise, done by Sumitra Bhave and Mohan Agashe on our research. And it's about improving nutrition in pregnancy. It's on YouTube. You are very welcome to see that. And finally, thank you to a large number of people, including our Sneha, that is DOHAD Indian Society. Then the participants, when they were young and now they are young people like this, the department staff and the village staff, and Pallavi, my wife, who has actually driven all the administration of this. Thank you.
Thank you so much, sir. This was indeed a journey that we traveled with you for the last half an hour, and that was wonderful. We have lots of questions, and I'm sure there's going to be a very active panel discussion, but we will do that in the panel discussion. We will hear Dr. Adrian, who has joined us. Welcome, Dr. Adrian, sir. I will introduce you to our audience so that, yes, just give me 1 minute. So Yes, Dr. Adrian is a professor of medicine in the Mayo Clinic College of Medicine and has been on the staff of Mayo Clinic, Rochester since June 2001. He is a graduate of University of Malta Medical School 1993. Currently, he directs a program funded by the National Institute of Health aims at understanding the pathogenesis of prediabetes and factors affecting islet cell function, which is what he is going to address today. His clinical interests include the diagnosis and management of hypoglycemic disease. This is wonderful. Thank you so much, sir, for sparing time to be with us. Welcome to IDEC Cafe, and over to you.
Thank you very much for having me. I'll start my screen sharing. Okay, and we can head off. So as you said, I'm going to be talking about the pathogenesis of prediabetes. And you could say that the inspiration for my research career is summarized by this slide. So this was a paper published by a colleague of mine at the time, who basically looked at Olmsted County, which is the county in Minnesota where the Mayo Clinic is located, and which actually has a unified medical record. So although there are 2 medical centers in the whole county, the medical record is common to both, and we can trace people all throughout their medical care. It also is characterized by strong population stability. So 95% of the population have not moved in 2 decades. Be that as it may, Dr. Deneen actually was out looking to see how the criteria, the recently changed criteria for the diagnosis of diabetes, might affect the incidence and prevalence of diabetes in Olmsted County. But the side effect of this publication was to show us that over a 10-year history, people with a fasting glucose less than 100 milligrams per deciliter had about a 5% risk of progressing to diabetes. People whose fasting glucose ranged in the 100 to 110 milligram per deciliter range had a 10-year risk of around 15%. The people whose fasting glucose was greater than 110 milligrams per deciliter, however, had a 40% risk of progressing to diabetes, which is a high number. But you could flip that number on its head and say 60% for various reasons do not actually progress to develop type 2 diabetes. And that heterogeneity was what intrigued me and led to my career, my research career at least. Anyway, when we come to talking about the defects of type 2 diabetes, my mentor, Dr. Riza, always used to show this slide, which was generated from his own data, but which illustrates the pathogenesis of type 2 diabetes. And in actual fact, people with diabetes differ from non-diabetic subjects because their fasting glucose is higher than normal subjects. After a meal, glucose goes higher and stays higher above fasting concentrations for a lot longer than non-diabetic individuals who usually within 90 minutes have returned to baseline. This difference is partly due by a delayed and decreased insulin secretion in response to glucose. And also of note, although fasting insulin is higher than that seen in non-diabetic individuals, for the prevailing glucose concentration, it is clearly inadequate. These defects are compounded by the fact that in normal subjects, glucagon typically suppresses for the first 90 to 120 minutes after a meal. In people with type 2 diabetes, glucagon doesn't actually suppress. In fact, it may paradoxically rise. So the net deficit and delay in insulin secretion compounded with impaired suppression of glucagon actually contributes to postprandial hyperglycemia. And in fact, I was always encouraged to think of the pathogenesis of prediabetes in terms of, first of all, insulin secretion. So insulin is released into the portal circulation and subsequently makes its way across the liver and is delivered to the systemic circulation. Now, across the liver, there is some degree of hepatic extraction. Whether that is an active or a passive process to this day remains uncertain. But the fact of the matter is that it affects systemic delivery. I do not think that to date there is sufficient evidence to suggest that changes in hepatic extraction can actually contribute to the pathogenesis of prediabetes, but this is still an area of active investigation. And then insulin is secreted. Insulin arrives into the circulation and affects insulin-sensitive and relatively insulin-insensitive tissues. But its actions are dependent on insulin action, which is the ability of insulin to stimulate glucose uptake and to suppress glucose production. There's also glucagon suppression, as I just showed you. And then there's a parameter which is often ignored, which is glucose effectiveness, which is the ability of glucose itself to stimulate glucose uptake and to suppress glucose production, which essentially means glucose release into the circulation by the liver, although the kidney provides a small percentage of glucose production. It's relatively small, and functionally, we're talking about the liver. The final process or parameter which modulates glucose delivery to the systemic circulation is gastric emptying. So when we come to talk about beta cell function, I always like to show this slide because ultimately it's useless talking about insulin concentrations. It's useless talking about insulin secretion. The insulin secretion has to be appropriate for the degree of prevailing insulin action. And in fact, Concord and Ark Royal have the same engines and the same number of engines. One breaks the sound barrier, the other one barely does 35 miles an hour. And the reason for this, despite having the same power, is obvious. So in a sense, it's important to appreciate that to assess beta cell function, you actually need to express insulin secretion as a function of the prevailing insulin action. One small note, because of hepatic extraction, you can't really use insulin concentrations to actually accurately assess insulin secretion, since insulin concentrations in the peripheral circulation reflect both secretion and hepatic extraction. Now, the relationship between insulin secretion and insulin action was first expressed as a disposition index by my colleagues and friends, Dr. Bergman and Dr. Cobelli. And in this particular example, you can see on the x-axis, there's insulin sensitivity. On the y-axis, there's insulin secretion. And over here, you have a normal subject who, during pregnancy, becomes insulin resistant, but because she can secrete more insulin, remains on this hyperbolic curve of insulin secretion and insulin action. Postpartum, insulin resistance decreases or insulin sensitivity increases, and insulin secretion can decrease appropriately as she travels down the hyperbolic curve. In contrast, somebody who is going to develop gestational diabetes in the third trimester, as measured here, you can see that there is some more insulin resistance compared to the other individual, but also for that degree of insulin resistance, she is secreting less insulin than the normal control. As postpartum insulin sensitivity decreases, insulin secretion also is decreased appropriately. However, it remains, the disposition index remains impaired compared to the normal subject without a history of gestational diabetes. So this relationship is what is used to quantify beta cell function and represents the disposition index. And here's another example where there was a therapeutic intervention. This is from the tripod study where Tom Buchanan was the lead investigator. And you can see that pre-treatment, people are shown in solid circles. With the advent of troglitazone, insulin action, which is the x-axis, increases, and there is a relative unloading of the beta cell, a relative decrease from baseline of insulin secretion. So people move along the lower part of the disposition index curve. However, it's important to appreciate that there are some limitations to the disposition index. And this here is work by Eli Ferranini and Ralph DeFronzo. Now, it must be said that insulin secretion was measured using an OGTT. And in actual fact, they are using a qualitative measure, which is the change in insulin concentrations divided by the change in glucose concentrations versus the M value, which is measured on a separate day by a clamp. So these are qualitative values, okay? They are different from what we're measuring. But you can see there is a hyperbolic relationship for normal individuals. There is a hyperbolic relationship for glucose intolerant individuals, but people with diabetes, the relationship here seems somewhat flat. And the important thing to appreciate is that across the spectrum of insulin secretion and action, in our hands at least, each dot, by the way, represents an individual, insulin secretion and insulin action decline in concert. As one declines, the other one also declines too. So across the spectrum, there isn't, insulin secretion rising to compensate for decrease in insulin action. Everybody seems to impair both parameters simultaneously. And if you look, normal fasting glucose, normal glucose tolerant individuals live on this hyperbolic relationship. People with impaired glucose tolerance live to the left and below of that relationship, and people with frank diabetes are even further down and to the left. So we started to worry that the disposition index actually may not may not properly partition the secretion and the action components of beta cell function. And so we thought that perhaps people have different hyperbolic relationships and move along different parts of the hyperbolic curve. So we thought that perhaps we would stress them acutely and see whether we can identify subtypes of prediabetes with different relationships of insulin secretion to insulin action. The hyperbolic relationship, by the way, is quantified by this function alpha. So it isn't just a straight multiplication of phi, beta cell responsivity, or beta cell secretion by SI, insulin action. It's actually to the power of alpha, which most of the time we use a population-based value for in our calculations. Now, this was given some added impetus by the description of the association of transcription factor 7 like 2 with type 2 diabetes by my friend, Dr. Struan Grant. This was work done in the DECODE cohort in Iceland, but basically TCF7L2 confers increased risk of type 2 diabetes, and to this day remains the common variant most strongly associated with risk of type 2 diabetes. And if you have 2 copies of the diabetes-associated allele of TCF7L2, your relative risk for diabetes increases by about to about 2.2 to 2.24. And this is the first time I'm going to mention the diabetes prevention program, which had shown previously that lifestyle is superior to metformin or placebo in preventing progression to diabetes. My friend, Dr. Jose Flores, was able to show that if you had 2 copies of the T allele, the diabetes-associated allele, in this cohort, your risk of progressing to diabetes was faster than the people with 1 allele, the CT genotype, or no diabetes-associated alleles, the CC genotype. And if you analyzed all treatment groups together, that relationship held. What was more exciting at the time was that using qualitative measures of insulin secretion, okay, the insulin to glucose ratio in this case, or the corrected insulin response, which is the delta insulin divided by the delta glucose, the people with the T allele had decreased beta cell function compared to people without the T allele. And in fact, we sought out, using the experiment I've just shown you before, to determine whether people with the TT genotype might have a different relationship between insulin action and insulin secretion than people without. In actual fact, we do not see that there's a different relationship. We just see that the relationship is shifted slightly to the left, so that basically at a given degree of insulin action at the lower end of the spectrum, people with the TT genotype secrete slightly less insulin compared to people with the CC genotype. And if we make them acutely insulin resistant by raising free fatty acids in the circulation, there's absolutely no difference in beta cell function in that case. Everybody collapses to the same relationship. By the way, it's important to emphasize each line represents 60 subjects with the TT genotype and 60 subjects with the CCC genotype. They are matched for age, they're matched for weight, and they're actually matched for fasting glucose status as well as sex. So these people were very, very closely matched as much as possible, the main difference being their genotype. But what was more striking than the small effects on beta cell function that we observed and had been described previously by people like Jose Flores, we actually observed that these people with the T allele also had a significant impairment in suppression of glucagon function, of glucagon post-meal. So here we had, for the first time, genetic evidence that genes associated with diabetes risk actually affect alpha cell function, implying that alpha cell dysfunction is likely an early mediator in the pathogenesis of prediabetes. And contrary to popular belief, alpha cell function does not seem to be inversely related to beta cell function. Insulin secretion does not seem to actively restrain alpha secretion. This is our cohort. Again, each circle represents an individual. And the lead investigator was Dr. Anu Sharma in this experiment. And we, like several other cohorts before us, again showed that beta cell secretion has nothing to do with either fasting glucagon or the glucagon response post-OGTT in the spectrum of prediabetes. We also showed that acute insulin resistance by raising free fatty acids actually raises both fasting and nadir glucagon in response to a meal. And you can see the relationship is more marked in the presence of FFA elevation, with the inverse relationship of glucagon with insulin action. Now, there was also something else in an independent cohort. We had previously also shown that in response to hyperglycemia, the TT genotype shows that alpha cell do not suppress glucagon in response compared to other genotypes, who again were weight and age matched. But we still hadn't shown whether alpha cell dysfunction can directly contribute to progression of diabetes. So as a sort of preliminary proof of concept experiment, at this time, we had already completed about 300 frequently sampled OGTTs, and approximately 7 years later, we were able to get 73 of them back in response to a questionnaire to repeat the OGTT. And we actually were able to show that fasting glucagon is associated with a decline in beta cell function, independently of baseline beta cell function, independently of age, independently of weight, and independently of sex. Now, this was a relatively weak effect, but the fact of the matter remains that, again, alpha cell dysfunction seems to lead to a subsequent decline in beta cell function over the period of observation. So I've shown you defects in glucagon secretion. The other question is there any heterogeneity in glucagon action, in the response to glucagon? And this is an experiment that we published approximately a year ago now, where we studied people on two occasions. On one occasion, we allowed glucagon to suppress normally, as would be expected after a normal meal. On the other occasion, we kept glucagon constant. But basically, the groups of subjects were divided according to the insulin dose they received. One group received 100% of the expected postprandial insulin dose. Another group received 80%. So you can see there's a decrease in the peak of insulin concentrations and a 20% decrease in area under the curve. And the final group received 60% of the insulin dose this group received. And again, you can see a decline in both the area under the curve and peak insulin concentrations. And these people were very well matched for age, weight, and sex. And you can see that in each group, there's a delta in the suppression of endogenous glucose production. But that delta was actually unaffected by the group they were in. So the response to a lack of glucagon suppression was heterogeneous across each group, but that heterogeneity was the same. So the actual delta was unaffected by the prevailing insulin concentration. And this heterogeneity seems to be unrelated to insulin action. It seems to be unrelated to weight. It seems to be unrelated to adiposity. So at the present time, there are no demographic or anthropometric characteristics that can allow us to predict the individual hepatic response to glucagon. So that's likely another source of heterogeneity in postprandial glucose regulation. In addition to secretion, we're actually going to have to start talking about defects or increased sensitivity to glucagon at the hepatic level. What could modulate this? Well, one concept which is likely quite important is the fact that at least in rodents, amino acid concentrations in the circulation directly affect alpha cell mass. Now, obviously, rodents are not humans. Rodents turn over their islet cells very quickly, and you can clearly understand that a change in amino acid concentrations could change alpha cell mass. That's unlikely to be the case in humans, but we can hypothesize that amino acid concentrations actually change alpha cell function. The point of the matter is that alpha cells secrete glucagon, and glucagon acting on the liver increases amino acid catabolism, which would be expected to decrease circulating amino acid concentrations. So in a sense, this alpha cell hepatic axis shown in rodents likely also to some extent exists in humans, and there is ample evidence to show that amino acid concentrations in the periphery can affect glucagon secretion. The question is whether glucagon action on hepatic amino acid catabolism differs in humans across the spectrum of prediabetes, and that is an area of active investigation. One other important point is to remember that concentrations do not equal secretion. And so we've actually developed a model where we can deconvolve glucagon secretion from circulating concentrations. And in fact, in this example that we show accompanying our publication, these are two individuals who at least appear to have the same degree of glucagon suppression. However, if you deconvolve concentrations into glucagon secretion, you can see that the rate of appearance of glucagon in the peripheral circulation differs quite significantly between the two subjects. So again, this will allow better phenotyping of affected abnormal individuals. The ADOPT study was another example of treatment of early type 2 diabetes post pre-diabetes. But again, it shows that insulin sensitizers are probably a little bit more successful at preventing progression of disease than insulin secretagogues. The point to be made is that in pre-diabetes, there have been numerous studies using insulin sensitizers. So this is troglitazone from the tripod study, which I previously showed you by Tom Buchanan. And you can see that basically using the minimal model indices, troglitazone treatment decreases insulin action, increases insulin action and therefore decreases insulin secretion. However, post discontinuation of treatment, everybody returns back to a decrease in insulin action with no compensation in beta cell function. So with troglitazone, the results do not seem to be durable over time. And you can see that even within a few weeks of cessation of troglitazone, glucose intolerance is again beginning to set in. And the same applies for pioglitazone. It's effective while on treatment, but once treatment is discontinued, the effects wear off. This is the DREAM study using rosiglitazone for the treatment, for the prevention of progression to diabetes. And you can see that people with rosiglitazone have lower rates of progression compared to placebo. Again, the effect wears off as the rosiglitazone is discontinued. But it's also important to point out that this benefit at sort of treating diabetes as it happens came at the cost of congestive heart failure, an increased incidence, as well as macular edema. And metformin in the diabetes prevention program, again, was effective in preventing the progression to diabetes It was not really associated with significant adverse events, but it did not change the natural history of progression. So when metformin was stopped, essentially these people started behaving like they were in the placebo arm and converting to diabetes at the same rate as the placebo arm. This was the phenomenon that they called masking, which is, in a sense, metformin was masking the development of diabetes. Perhaps a better way of looking at it is to suggest that metformin was treating diabetes as it happened in the cohort receiving metformin. So I'll conclude by saying that islet cell function is the primary regulator of glucose metabolism in humans. Insulin secretion and signaling is an important contributor to pathogenesis of type 2 diabetes, but abnormal glucagon suppression is an early change in the pathogenesis of type 2 diabetes. So that in the future, when measuring beta cell function, it will be also important to consider the effect of alpha cell function on the response to oral glucose. I'll stop by acknowledging my study subjects, the people in my lab who have contributed to a lot of these studies, as well as my mentors and my collaborators at the University of Padova. So thank you for listening to me. I'm sure there will be questions.
Thank you so much, Dr. Adrian, and I'm sure there'll be lots and lots of questions. And I hand over to Dr. Neeta Deshpande now to take on a panel discussion with Dr. Adrian and Dr. Yadnik. Over to you, Neeta.
Yeah, thank you, Archana. This has indeed been a treat for all of us listening in over here and for the 2,500 people who have logged in. So I'll just straight away kick off the panel discussion by asking my first question to Dr. Yagnik, sir. Sir, this is a question of my own. I'll take up questions from the audience as well. My question is, the current epidemic of diabetes that you see in India today and have been seeing for the last decade or so, what is the contribution of undernutrition versus overnutrition?
Okay, thank you, Neeta. I think as a country, if you look at the birth weight of Indians, even in the NFHS 5, which was done over last 5 years, the average birth weight is 2.8 kilograms. And by international standard, up to 20 to 25% of babies are small for gestational age. Larger babies are rare. So intrauterine undernutrition is still quite common. Intrauterine overnutrition is now coming from young girls who themselves were undernourished in their early life and therefore have a so-called gestational, but as I showed you, that gestational thing is present from very early age. So it's a combination of undernutrition, which is multi-generational,
Yes.
with a rapid lifestyle change since 1980, which is combining into causing an explosion. And we saw that 2017 report of prediabetes in 5 to 9-year-old children and to 10 to 19-year adolescents. And many states have a prevalence more than 20%. So I think it's a multi-generational undernutrition with rapid transition that
Yeah.
Ganesh, which I showed you,
Yes.
instead of traveling on the back of the mouse, he is now clicking the mice of the computer.
Right. Thank you. Thank
And
you.
also I showed it for liver growth,
Right.
poor liver growth in utero, which is a surrogate probably for pancreas and other organs, and subsequently larger liver. And actually the larger liver at 18 years, only 2 out of 700 had what was diagnosed as fatty liver. So it was a pre-fatty liver.
Right. Okay.
Yeah.
So I'll come back to that in just a bit. I would like to ask Dr. Vella one more question. You said that genes associated with diabetes risk also affect alpha cell function. With that as the background, what do you think then is the role of DPP4 inhibitors versus metformin considering that DPP-4 have effect on the alpha cells and glucagon. So considering that, do you think that can also be used to prevent diabetes, or is it only metformin?
So there isn't a lot of good data using DPP-4 inhibitors in prediabetes. To lower glucose in prediabetes, you need to have glucose above a certain threshold, since the insulin secretion stimulated by DPP-4 inhibitors is glucose dependent. So clearly, the more abnormal both fasting and postprandial glucose is, the more likely there will be some effect. In terms of lowering fasting glucagon, DPP-4 inhibitors have had heterogeneous reports. I actually don't think it's particularly strong, the effect on fasting glucagon. There are some studies which show there is, and there are some studies which show that there isn't. The point is, however, that the assays that were being commonly used to measure glucagon at the time, so this is a decade ago, would actually also measure other pro-glucagon fragments. So we don't really know whether that lowering came at the expense of glucagon per se, or some other contaminant, some other pro-glucagon fragment like glycantin, which cross-reacted, especially to the old millipore assays. But the point of the matter is there isn't much data in support of DPP-4 inhibitors in the use of prediabetes.
Right. So what we have now and what we have data for is metformin. Considering that in India today, we recently received in the last couple of months a DCGI regulatory approval for use of metformin in prediabetes. And we also have the data for it. So I suppose that is, we talk about metformin at this point of time.
Yeah, so I mean, in the greater scheme of things, as I said, metformin doesn't really change the natural history of progression. It just treats the diabetes as it happens. But be that as it may, metformin has been around for a long time, and we really know its long-term safety profile. So I think it's perfectly acceptable to use it, recognizing that aggressive lifestyle intervention is superior and aggressive weight loss is superior in preventing progression to diabetes. It still has a role to play. And also it's a safe medication. Right. On the other hand, if you're balancing decreasing the progression rates temporarily versus, admitting 10 patients per year with congestive heart failure due to fluid overload from a thiazolidinedione, I think the risk-benefit ratio is against using thiazolidinediones in prevention of progression of prediabetes. But metformin, I will accept that risk-benefit ratio because it's very favorable.
Right.
So, Nita, there is a point about Indian diabetes prevention trial.
Yeah.
Where metformin added to lifestyle did not improve did not reduce the rate of progression. The dose used was 250 milligrams twice a day.
Yes.
That could be one problem, but at least some effect should have been there. So there was no effect in the Indian diabetes prevention trial of metformin.
Right. So that
over and above the lifestyle.
Yeah, so that probably give credence to your what you said about the secretion problem being more important. Yeah. So now, Dr. Yagnik sir, I'm taking up a question from the audience, and the question is, pre-diabetes is so heterogeneous. Evolution of pre-diabetes has to be very complicated. Can you simplify it for us?
See, broadly speaking, like what Leif group did, he did subgroups of type 2 diabetes and came up with 5, of which one was the GAD antibody positive. So it was the autoimmune type. I teamed up with Leif and Rashmi, and we published in Indians the 4 subtypes which they had described in the Swedish, and I showed the comparative slide. Okay, in the Swedish, those who had developed diabetes before 45 years of age, 2/3 were the obesity related, while in Indians, it was the insulin secretion defect. Interestingly, in Indian women in our study, they had obesity-related diabetes more common, just a little bit more common than the insulin deficient. And I think now we are building up evidence in our cohort because the girls, as I said, married, they became pregnant. So we have a glucose tolerance in pregnancy. And we had additional glucose tolerance when they were for subsequent follow-up. And what we have realized is that these thin girls have substantially retained weight after pregnancy. And therefore, if we then look at their mothers, again, whom we have serially followed up, they were short and they have developed obesity very rapidly after 35 years of age. So in other words, the early life scenario in India is one of undernourishment, especially in the reproductive age, certainly in the villages, and subsequently combined with overnutrition. So it becomes confusing for the average doctor. And therefore, I think simplest thing to do is to document simple clinical things properly. Of course, measure height and weight, we take it for granted. I don't know how many primary health centers do it. Calculate BMI. I am a great fan of measuring body fat. So if we use a caliper to measure subscapular skin fold, all our research about adiposity ultimately boils down to subscapular skin fold. That tells you about adiposity. And then finally, if you measure glucose and either insulin or C-peptide in the fasting blood, you can model it and then classify them. There will be people who were predominantly deficient. There will be people who were predominantly resistant and with a combination in between. And we need Indian trials. We are deriving all our consensus based on, like, say, prevention trials in people with an average BMI of 35, while even in the Indian diabetes prevention trial, the average BMI was 25.
25.
I think there's a great need for our data. And I think if RSSDI and other organizations, we all get together and do it, it will clarify things for us. Dr. Vela showed the sigmoid curve, and we have now sent a paper for the sigmoid curves in those who were pre-diabetic at 18 years, how that has progressed from 6 years to 12 years to 18 years. And it's an extremely interesting picture. If you allow me, I can show it, and we can also take Dr. Vela's sort of comment on that, because his studies are mostly in adults and diabetic compared to NGT. This is an evolution in the age, and I can show you something quite interesting. Just a second. Are you able to see?
No. I think your screen is not shared.
No?
No.
Okay, I'll share again. Now?
Now
Now
yes.
it's here.
Yeah. So like, so these people have been divided as NGT and pre-diabetes on their oral glucose tolerance at 18 years, but we had similar data at 12 years and 6 years. And these are same scales for HOMA beta and HOMA S, because that was an epidemiological study and that's what we had. See, if you start at 6 years, there is a very wide spread on the sensitivity axis. And the beta cell function is quite low. So these people are quite sensitive as a group at 6 years. By 12 years, the x-axis has started contracting and the y-axis is growing. So that's the compensatory beta cell secretion. And by 18 years, if you see the sensitivity is now below 400, and the beta cell function has gone up to beyond 200 or 250 units. And at all points, those who were pre-diabetic at 18 years, which is the red dots, as compared to NGT, which is the green dots, they are on the left of the green dots. So it starts very early and progresses, and arbitrarily we call them pre-diabetes, etc. in the adult age. In children, we really don't have any criteria. That's why we divided them into quartiles or tertiles. And this actually shows nicely on this plot where we put log against log. And if we put the NGT curve of sensitivity against beta cell function, it's tracking like this. And pre-diabetes are always tracking at a lower. And this is right from 6 years of age. I dare say if we had done a test at 2 years of age, we would have probably found the same thing. And the higher glucose, which I showed, I am now a DOHAD person, and periconceptual programming is very important. And therefore, especially for girls, having these high glucose and other abnormalities from childhood affect their ova and the periconceptional embryo exposure. So I think that's a very strong message which I got from what we have done over years. And therefore, to me, like Dr. Vela was saying, primary prevention of diabetes is actually treating high glucose either by lifestyle or by tablets. And as soon as you stop it, you again go back to where you started. And rather calling it primary prevention, I think is quite a misnomer. It's quite misleading.
Right. Dr. Yagnik sir, according to what you said, that when we start to try and treat pre-diabetes, when we start to try and intervene at the level of pre-diabetes, we have probably already missed the bus, is what you would say. So in that case, if we were to start at 12 years or 6 years, what is it that we could possibly do at that time? If we really
given the current ideas and the safety of the lifestyle intervention, that's what we should do. But because using drugs at a young age without knowing the cost benefit or the cost effectiveness, etc., is not possible. And therefore, in a way, the whole sort of thing is that we have come to this stage after many generations of problem. We are not going to come out of it in just overnight by treating in one generation. And therefore, we have to prepare to prevent the problems for the next generations. And therefore, improving the health of the adolescents as early as in the life as possible is going to help reduce the programming of these in their offspring. So what we could do is improve their nutrition, improve their physical activity, reduce the stress, improve the sleep patterns, whatever.
Right. So my next question I'm taking to Dr. Adrian. Dr. Adrian, you spoke about the alpha cell hepatic axis. So could you elaborate a little more on that and the clinical implications of it?
Sure. So for years, it's been known that branched chain amino acids, if elevated, are predictors of risk for type 2 diabetes. But mechanistically, they have no direct toxic effects on beta cell function or islet function for that matter. Now, as it so happens, we know that amino acids in the circulation can affect glucagon secretion. If you give arginine IV, it is well known that in addition to stimulating insulin, you will stimulate glucagon secretion. That's well known. It's also well known that glucagon and its actions on increasing glycogenolysis and supporting gluconeogenesis are maintained in diabetes. In fact, it's part of the mechanism of hyperglycemia. However, now there is some evidence, a couple of human publications, where there is heterogeneity in the response of amino acid catabolism to glucagon. So people with hepatic steatosis, for example, seem to be resistant to the actions of glucagon in causing amino acid catabolism. So you can envisage a scenario where partial resistance to glucagon's actions can raise circulating amino acids, which in turn raise circulating glucagon concentrations, which in turn cause hyperglycemia through glycogenolysis and gluconeogenesis. So there are clinical implications if this is true. It's certainly true in animals. Whether it's true in humans is the subject of active research at the present time. So, so that's what we're actually actively seeking to establish. But the point is that it is clear that circulating concentrations of hepatic metabolites are likely to directly affect glucagon secretion, which in turn can affect hepatic metabolism of said precursors of those metabolites. And that's the alpha cell hepatic liver, hepatic axis in summary, essentially, and how it may play a role in pathogenesis of diabetes.
Right.
So Dr. Vela, is there any role for branched chain fatty acids?
It's an excellent question. So until recently, it was thought that glucagon has very little effects on lipolysis, on hepatic lipolysis and lipogenesis. Now, there is a little bit of evidence suggesting that there are small effects of glucagon on de novo lipogenesis, for example, and on lipolysis. And certainly, if you block the glucagon receptor therapeutically or attempted therapeutically, all pharmacotherapy based on glucagon receptor blockade has been abandoned because even in humans, hepatic steatosis develops. So clearly, there are actions of glucagon on lipolysis and de novo lipogenesis. What the magnitude of those effects is uncertain, and what the stimuli are for glucagon secretion and glucagon action are still very uncertain. But it's, again, an area we actively are pursuing.
See, we are pursuing vitamin B12 deficiency as a possible contributor because in the Pune study, we found low maternal B12 and high folate predicted higher adiposity and higher insulin resistance in the child at 6 years. B12 has effect on two enzymes. One is methionine synthase, which feeds into folate and the methylation cycle, which has to do with DNA synthesis and methylation. And the other enzyme is mutase. And mutase helps propionic acid to be converted to succinic acid. If B12 deficiency, mutase is not working, the propionic acid accumulates and is converted to methylmalonic acid.
Sure.
And a number of other branched chain amino acids, branched chain fatty acids. And that itself actually prevents transfer of propionic acid into mitochondria, affecting the beta oxidation. And we have written this hypothesis in 2008 in Diabetologia when we published the effect of low B12 in the mother on the insulin resistance and adiposity in the child. And you are saying same mechanisms from your viewpoint, which we are describing from an undernutrition of vitamin B12 side. And if you go back in literature, there are any number of publications in 1950s and 60s where childhood undernutrition and kwashiorkor was thought to be a major cause of hepatic steatosis.
Sure.
So it's quite amazing that today we take NAFLD as an overnutrition disease. And previously it was an undernutrition disease. And now again, like I said, from combination of a multi-generational undernutrition and recent overnutrition, we are actually revisiting these phenomena.
Yeah. So the question is actually whether hepatic steatosis itself is a marker of impaired glucagon action at the liver level. Again, a phenomenon that we are actually studying.
Yeah, I mean, we wrote a number of papers again in 1990s in collaboration with George Alberti and Derek Ockaday, with whom I trained. And it was using the Krebs sort of phenomena. And we infused intralipid and other things and showed the malnutrition related diabetes to be ketosis resistant.
Yeah.
Again, indicating there was problem handling the fatty acids after they entered the mitochondria. Today, I have a suspicion that these patients had severe B12 deficiency, which was contributing to that. So again, I mean, actually nutrition, especially of micronutrients and the mitochondrial function, seems to be a common denominator here.
Sure. Full disclosure, as a medical student, I spent a month with George Alberti a long time ago. So.
Okay. Yeah, George is a mentor, friend, and everything. So yes, I mean, I met him first in 81 and I did 3 years in Oxford.
Sure.
Right. That's really interesting to know. And just a last couple of questions, because now we are coming to the end of our time. This question, either one of you can answer. And the question is there any role at all of vitamin D deficiency in the pathogenesis of prediabetes or diabetes?
So perhaps I'll take a stab at it. I think obviously it depends on how you define vitamin D deficiency. Nowadays, at least in the Western world, the thresholds for vitamin D deficiency have been moved up and up and up. And certainly, you know, vitamin D deficiency with osteomalacia described in the textbooks as recently as 20 years ago is a very different phenomenon to what fellows in continuity clinic come to describe as quote unquote vitamin D deficiency. Certainly at the higher end of vitamin D concentrations, there is very little evidence that there's direct effects on islet function in humans. And there's not much to support defects in insulin resistance and insulin signaling at those levels. Not very well studied at the lower ends of vitamin D concentrations. But again, I would suggest that it's an academic question in the sense that there are very good reasons to treat vitamin D irrespective of the prevention of diabetes.
Right.
Especially at those lower ends.
So Neeta, again, we have a story in Pune maternal nutrition study that in addition to low B12 and high folate, low maternal vitamin D in pregnancy was a predictor of subcutaneous adiposity in the child at 18 years of age.
Okay.
Now, these are all observational but interesting intergenerational studies. And there has been a trial of vitamin D in Southampton, and they are following up the children, and they will see whether what we found as observational, whether in trial that has been reversed in any way.
Right. So this has been a very interesting session indeed. And before I wind up, I would like both our faculty, both Dr. Adrian and Dr. Yagnik, to give one crisp message each as a take-home message to the physicians who are listening in on what they should be doing from tomorrow in their practice with respect to what they've heard today.
Well, I think if one is to focus on therapy in pathogenesis of prediabetes, I think the message would be first and foremost lifestyle. Pharmacotherapy may have a role, but at the present time is inferior to the benefits of lifestyle.
Right. Dr. Yagnik, sir?
I would say if you want to prevent pre-diabetes and diabetes at community level, it's never too early, never too late.
Right.
And second message, which we popularized in 2008 and 9 as Kathmandu Declaration, is that women's health is nation's wealth. So that's what actually I would suggest, that we concentrate more on the health of the young women.
Right. Thank you. Thank you very much, sir. And thank you very much, Dr. Adrian. Real pearls of wisdom. Thank you so much. I'm handing it back to Archana to do the concluding formalities.
Thank you, Neeta. Thank you so much. This was an exciting panel discussion, so much to learn and so much to take home. I would like to invite our colleague, Shilpa Joshi, to have a formal vote of thanks and a message that she has for all of us. Over to you, Shilpa.
Thank you so much, Dr. Archana. And after an interesting and a thought-provoking evening, it's time to say thank you to everybody. So thank you, Dr. Vela and Dr. Yagnik, for a great thought-provoking evening today with great crisp messages. I think in the IDEC committee, we are 3 women. And if you say that, women's health is nation's wealth, then it means a lot to all the 3 of us. So thank you, sir. And, we had an audience of 2,500, which just goes to 2,800, I'm sorry. So that just goes to show how interesting this evening was. So on behalf of Team IDEC, I would like to thank everybody in the audience who have spent their time to be here today evening. And we also thank USV for all their support. I take this opportunity to invite all of you to the IDEC meeting, which is going to happen on 5th, 6th, and 7th August 2022 at Pune. So please save your dates, and you're all invited. Thank you so much. Over to Dr. Archana.
Thank you, Shilpa. So I think it has been a wonderful evening. We are all enlightened and we are looking forward to the next Wednesday Cafe. This is usually going to be the 4th Wednesday of every month. We would love to have audience feedback. We would love to have your questions, which we can hopefully share with our speakers and get back to you. Thank you so much. Thank you, everyone. Wishing everyone a good night.
Good night, everyone. Bye.
Thank you.
Thank you. Bye-bye.
Bye.
Bye-bye.
We are offline.
Thank you so much. We are offline.
Right.
Great show, guys.
Oh, that went well. Where is?
Oh.
Are we offline?
Yes, we are.
Yes, yes, ma'am.
Yeah.
Yes, we are.
We can breathe.
Done. Second one done.
Yes. There weren't fireworks. Luckily, they actually agreed to each other.
Yeah.
But a lot of good stuff.
Yeah.
Very good stuff.
But I'll tell you one thing, Dr. Yagnik was mind-blowing.
I know. He was very good today.
And the quality of his work is par excellence. It's better than many of the Westerners also. It's amazing.
That's
Absolutely amazing.
Good show. So now we rest easy. Start work for IDEC meeting.
Yes, that's on. Right.
Let's breathe.
Let's sleep.
All right, get after you guys.